We're doing macromolecules, identifying them, and we can see across the bottom we have 4 different tests. So we have a test for carbohydrates with 2 different tests we can do. We have a test for proteins, and we have a test for lipids. So if we look down to the Gizmo warm-up, have you ever wondered what is in your food? Scientists use a variety of tests to determine the nutritional content of food. You will learn 4 of those tests with the identifying nutrients Gizmo. So below the food samples label, so food samples right here, drag test tube A into the food sample holder. So I'm just going to take A, put it in here, okay? And then it says below the Benedict test, click the test button. What is done in the Benedict test? So here's the Benedict test. I'm going to click test. So we just want to describe what is happening. So we put a couple drops of A into the test tube. We put a couple drops of the Benedict into the test tube. And then we also heated it. Okay, so we want to make sure that we include that in our information. For number 2, the Benedict test is a test for monosaccharides. So simple sugars like glucose and fructose, which is the sugar found in fruit. In contact with monosaccharides, the Benedict solution turns blue to orange or green. So does sample A contain monosaccharides? So the answer would be yes, because it turned into that orange color. So you would type yes into that spot. All right, let's click reset up top. Then it says drag sample A into the food sample holder, okay? So most food is composed of 3 different types of molecules: carbohydrates, proteins, and lipids. Carbohydrates such as starches and sugars are a major source of energy. Simple sugars are found in sweets and fruits. Starches are found in potatoes, cereal, pasta, flour, and other plant products. Proteins are used in body structures such as muscles, skin, hair. They are rich sources of proteins. Rich sources of proteins include meats, dairy products, and beans. And then for lipids, your fats and oils, they're used for energy, insulation, and as an essential building block of cells. If we remember back to our cell membrane, it was a lipid bilayer. Meats, dairy products, and oily plants such as olives are rich in lipids. So now we have to look, how do we test for carbohydrates, proteins, and lipids? So we just did number 1. Under the Benedict test, click test, does sample A contain monosaccharides? We already did that one, and it was yes. The answer was yes for number 1. So number 2, let's test the Lugol. Okay. So test for a polysaccharide. We're going to do this test. So we're going to click test for Lugol. And then it says in the actual number 2, it says iodine turns dark purple in the presence of starch. So this did not turn dark purple, so does it contain starch? You would say no. And then for number 3, the Biuret test. So this is our proteins test. Biuret's a solution to test for proteins. It'll turn purple when proteins are present. So here we have no protein, okay? It did not turn purple. And then for the Sudan red test, it's going to test the fats. Sudan red is to indicate the presence of lipids. When lipids are present, the dye will be absorbed into the lipids and will appear as concentrated spots of colors on the test tube. So let's test for that. Okay, so there are no spots, so it tested negative for lipids. And then for number 5, what macromolecules did sample A contain? The only one it contained was the monosaccharides, okay? So it contained carbohydrates. And then analyze what kind of food is sample A most likely to be. So we need to figure out, is it monosaccharides or carbohydrates, right? So which one of those 4 foods would be a carbohydrate? Go ahead and answer that one. For B, we're going to hit reset. And we're going to test all of these samples. So use the 4 available tests. That's to find the nutritional content of samples E, G, and M. So A has been done. We already did A, okay? And so let's do E. So if we drag E here, we're just going to test each of these. Okay, heated for 5 minutes. Okay, that's a negative test, so we would put a negative sign for the Benedict's test for E, put a minus sign. Now we're doing the Lugol test. This is also negative. Remember we said it would turn a dark blackish color if it was positive. Protein. Okay, that's positive. Proteins turn purple. You can always remember P and P. Protein, purple. And for the lipids, we're looking for red spots. If we don't see red spots, if it's all red, then it's a negative test. There we go. That's red spots. So it would be negative, negative, positive, positive. All right, let's reset and let's do G. Okay, that's a negative test. Let's test for the Lugol, for the starch. Also negative. Okay, we want this to turn black. If it turns black, that's a positive test. Test for proteins. Remember protein, if it's purple, it's a protein. That's a negative test. And test for the lipids. That's also a negative test. We were looking for all those little circles. We don't have any. So it's negative, 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 negative for sample G. Okay, let's do sample M. First we'll test the Benedict. That's a negative test. Let's test for the Lugol. Okay, that's a positive test, right? It's a dark color. They say black, but it could be considered purple. That's a negative test. Also negative. So we got negative, positive, negative, negative. All right, so you should be able to fill out the rest of that chart, and then you should be able to answer the two questions below the chart.